Okay, have you ever wondered how a thought even happens? Or how you form a memory? Well, it all comes down to these tiny, unbelievably powerful switches in your brain. And today, we're going to tear one apart and see how it works. We're talking about the excitatory synapse, the microscopic go signal that makes, well, everything happen.
So here's our game plan. First, we're going to meet the chemical messenger that actually carries that go signal. Then, we'll get to know the proteins whose job it is to catch that signal. After that, we'll see how two of these proteins work together in this really clever way that's kind of the secret sauce for learning. And finally, we'll see how this whole incredible piece of machinery is actually built and held together.
All right, so every good story needs a hero, right? Well, in the story of how your brain fires on all cylinders, our hero is a tiny little molecule that does pretty much all the heavy lifting for almost every circuit in your head.
So what is it? What is this master molecule? This chemical that's responsible for flipping the vast majority of the on switches in your entire nervous system?
It's glutamate. You know, this simple amino acid is the undisputed champion of getting things going in the brain. It's the primary voice shouting, "Fire! Pass the message along! Do something!"
So a question pops up pretty quickly. If glutamate is getting fired off all the time to send all these signals, what happens to it? Does it just build up? Well, no. The brain has this really elegant solution, a super efficient recycling program. And this cycle is just a perfect example of the teamwork happening at this tiny scale. So a neuron fires, it releases glutamate. But instead of just wasting it, these neighboring support cells, they're called glia, they swoop in and scoop it up. Then they do a little chemistry, change it into a safe, inactive form called glutamine, and then they just hand it right back to the neuron. The neuron then quickly converts it back into glutamate, ready to go again. It's an incredibly sustainable system. Just brilliant. Okay, so glutamate is the message, right? But for any message to be heard, you need a receiver. In the brain, we call these receivers receptors, and it turns out they come in a couple of different flavors. You've basically got two main families. First are the ionotropic receptors. You can think of these like a really simple, fast-acting gate. When glutamate hits them, they just pop open a channel and let ions rush into the cell. Boom, simple. Then you have the metabotropic receptors. These are a bit more dramatic, they're slower, and they kick off a whole biochemical chain reaction inside the cell. For our story today, though, we're going to focus on that fast-acting ionotropic family. Now, even within that fast-acting family, you've got three main players, and they're named after some lab chemicals that can activate them specifically. You've got AMPA, kainate, and NMDA. And while they're all doing important things, the two that really run the show at most of these excitatory synapses are AMPA, that's the workhorse for just fast, everyday signaling, and NMDA, which is the specialist, the one that gets involved in changing and strengthening these connections over time. So let's zoom in on these two key players, AMPA and NMDA. What's so cool is that they often sit right next to each other at the very same synapse, but they react to the exact same glutamate signal in totally different ways. So how does that even work? Right? How can one single chemical signal produce two completely different results at the same time? Well, it all comes down to the kind of electrical current that they generate when they open. And this slide really nails the key difference. It's all about timing. The AMPA receptor, it's a sprinter. It opens and closes in a flash, creating this super sharp, really brief electrical spike. The NMDA receptor, on the other hand, is a marathon runner. Its response is slower to get going, but it lasts for a much longer time, creating this prolonged wave of current. So why the different speeds? It all comes down to how sticky they are for glutamate. You see, AMPA receptors have a low affinity. Glutamate binds and then lets go almost instantly. It's like a quick handshake. But NMDA receptors have a high affinity. When glutamate binds there, it holds on tight for a lot longer, and that's what keeps that channel open and the signal going. And this long-lasting signal is a huge part of the NMDA receptor's most famous and honestly most important feature. It's not just a simple gate, it's a smart device. It's known as a coincidence detector, and this is absolutely critical for how we learn and form memories.
So what exactly is a coincidence detector? Well, just think of it like a switch that will only flip to on if two very specific things happen at the exact same time. It's literally looking for a coincidence.
For the NMDA receptor, there are two conditions that have to be met. First, glutamate has to bind to it. That's like putting the key in the lock. But here's the catch. Even with the key in, the channel is physically blocked by a magnesium ion. It's like a plug.
The second condition is that the neuron has to already be electrically active. That activity, that depolarization, provides the jolt needed to pop that magnesium plug out. Only when both things happen at once, the key is in and the power is on, does the channel finally open wide.
And that right there is the magic trick. Because it needs the cell to already be buzzing with activity from other inputs, the NMDA receptor can basically sense that something important is happening. It's the cell's way of saying, whoa, this signal is arriving at the same time as a bunch of others, this connection must be important, let's strengthen it. This is literally the cellular basis for how we learn. It's how neurons that fire together wire together.
Okay, so we've met our messenger, glutamate, and our two star receivers, AMPA and NMDA. But how are they all organized in the actual synapse? The answer is with incredible, mind-boggling precision. A synapse isn't just some random soup of proteins floating around. It is a highly engineered piece of molecular architecture.
Okay, so take a look at this. This is your classic excitatory synapse. Up top, you've got the terminal. It's packed with glutamate, ready to be released. And down below is the receiving cell, and you can see it's studded with those receptors we've been talking about. But notice, they aren't just scattered all over the place. Now, if we could zoom in, way in on that receiving membrane, we'd see this incredibly dense web of other proteins. This whole complex is called the postsynaptic density, and you can think of it as a kind of molecular scaffolding that holds all the key players exactly where they need to be.
The master organizer of this whole structure is a protein called PSD95. The best way to think about it is like molecular Velcro. It literally grabs onto the NMDA receptors and anchors them firmly in place. Then it uses this little adopter protein, called a TARP, to grab onto the AMPA receptors, clustering them right next to their NMDA partners. This ensures they are all in the perfect spot to respond to glutamine.
And here's the takeaway. None of this is by chance. The exact placement of these fast sprinter receptors and the smart marathon runner receptors held in place by the sophisticated protein scaffold, that's what makes your synapses so fast, so reliable, and so adaptable. The architecture is absolutely the key to its function.
And that really brings us to a final, pretty amazing thought. We've just taken apart the fundamental go switch in the human brain. And understanding this intricate molecular machine in this much detail, well, it opens up some incredible possibilities. I mean, if we can learn to fine-tune this system, to maybe adjust the number of receptors or strengthen the scaffold that holds them together, what could that mean for conditions where these circuits go wrong? Things like memory loss or addiction or learning disorders. The foundation for a whole new generation of therapies is being built right now, right here at the Synapse.